Ladies and gentlemen, members of the Faculty of Medicine, or invited guests, dear students, and our special guest, Professor Shimon Sakaguchi. It is my great pleasure and privilege to welcome all of you to the presentation of the ceremony of the Debrecen Award for Molecular Medicine of the Faculty of Medicine. The Faculty of Medicine has a long tradition in encouraging basic and clinical research. Our eminent research groups strive to reach the front line of biomedical research and to push the boundaries of science by pursuing cutting-edge research. Our faculty also aims to strengthen its position as a regional center for international graduate and postgraduate training. As an acknowledgement of our leading role in the region, the European Union recognized us as a center of excellence in molecular medicine. We also feel that in this capacity, we are obliged to identify and recognize researchers who contributed to the field of molecular medicine. The international prize, the Debrecen Award for Molecular Medicine, is offered to recognize groundbreaking, extraordinary and sustained contributions to biomedical research. The award acknowledges significant advances in the field of molecular medicine with demonstrated benefits applied to improved patient care. This year, the selection committee of the Debrecen Award for Molecular Medicine considered Professor Shimon Sakaguchi as one of the nominees for the award. Now, I ask László Csernok, Vice Rector of the University of Debrecen, to announce the decision of the 2023 Debrecen Award for Molecular Medicine.
Ladies and gentlemen, it is my duty and privilege to report that the professors of the Faculty of Medicine have decided by secret vote that the 2023 Debrecen Award for Molecular Medicine should be presented to Professor Shimon Sakaguchi. Professor Sakaguchi is an immunologist and a distinguished professor of Osaka University. He is best known for the discovery of regulatory T cells and to describe their role in the immune system. This discovery is used in the treatment of cancer and autoimmune diseases. Dr. Sakaguchi studies the molecular and cellular mechanisms of immunological tolerance and immune regulation. He has shown that a population of immunosuppressive T lymphocytes, designated regulatory T cells, are present in the immune system, and its deficiency or dysfunction is causative of a variety of immunological disorders, including autoimmune diseases. He has investigated the molecular basis of regulatory T cell development and function. In addition, Professor Sakaguchi and his laboratory have demonstrated that numerical expansion of regulatory T cells or strengthening of their suppressive activity is capable of preventing and treating autoimmune diseases and also establishing stable tolerance to transplanted organs, while their reduction in number or suppressive activity is able to provoke effective immunity against cancer. Professor Sakaguchi is currently investigating how regulatory T cells can be targeted in humans to control a variety of physiological and pathological immune responses in clinical settings. Now I ask Zoltán Pap, Vice Dean for Scientific Affairs, to highlight some relevant facts and data pertaining to the biography of Professor Shimon Sakaguchi.
Professor Shimon Sakaguchi obtained his MD degree in 1976 and his PhD degree in 1982 from Kyoto University, Japan, where he was also trained as a pathologist and immunologist. After performing postdoctoral studies at Johns Hopkins and Stanford universities, as a Lucile P. Markey Scholar, he served as an assistant professor in the Department of Immunology at the Scripps Research Institute, Loyola, California, United States. He returned to Japan in 1991 and continued his immunology research as the head of the Department of Immunopathology at Tokyo Metropolitan Institute of Gerontology. From 1998 to 2011, he was appointed as professor of Institute for Frontier Medical Sciences, Kyoto University, and served as a director of the institute for several years. In 2011, his lab moved to Osaka University. He was elected to foreign member of National Academy of Sciences, United States, in 2012, and has been awarded many international prizes, including Cancer Research Institute's William B. Cowley Award in 2004, Keio Medical Science Prize in 2008, Canada Gardner International Award in 2015, the Crawford Prize in 2017, the German Immunology Award, previously called the Avery Landsteiner Award in 2019, and the Paul Ehrlich and Ludwig Darmstädter Prize in 2020. He received an honorary degree from the University of Birmingham, UK in 2019, and the Order of Culture, the highest accolade for Japanese scholars, from the Emperor of Japan in 2019.
Dear Professor Shimon Sakaguchi, now you are kindly requested to come forward to this table for the conferment of the Debrecen Award for Molecular Medicine. Sakaguchi will now deliver his inaugural lecture. Congratulations. We can take that.
So, ladies and gentlemen, it is my great honor and pleasure to be here and awarded this year Debrecen Award for Molecular Medicine. I'm honored for the discovery of regulatory T cells and then its the characterization and then also application for treatment of the immunological diseases and cancer. So it is a great encouragement for me to receive this award for further continuing our research and then contribute to medical science. So I really thank the faculty of Debrecen Medical School for choosing me for this award. Thank you very much. So let me start my lecture. So the title of my lecture is Regulatory T cells for controlling immunological diseases. So basically, I'd like to tell you three issues. And one is perhaps the audience, many of them are not immunologists, so it may be good for tell you the history of regulatory T cells, how it was discovered, and how it has been studied. And second, how the regulatory T cells can be targeted in human diseases. So let me talk about, especially about the role of regulatory T cells in autoimmune diseases in humans. And third, and for the future, how we can target regulatory T cells to treat and then prevent immunological diseases. So what is important is how we can achieve antigen-specific immune suppression. So our immune system, it reacts with viruses and bacteria and protect our bodies. But it doesn't react with self-constituent. If it reacts and damage, you'll see autoimmune disease. And so this is non-unresponsiveness to self-tissue, self-antigen. It is called immunological self-tolerance. And if we understand the mechanism of immunological self-tolerance, we may be able to understand the mechanism of autoimmune diseases, how we can trigger immune responses against tumor cells, which have arisen from normal self-tissue. And the ultimate goal of organ transplantation, that is to make the immune system to treat the other's organ as if it is a self-organ. And allergy, well, so the many allergens, like say pollen, it's not a dangerous substance, but if it reacts with them, then you'll see allergy. And microbial immunity, for example, we do have commensal bacteria in the intestine, but we don't react with the commensal bacteria. If it reacts, you'll see inflammatory bowel disease. So to understand immunological self-tolerance, that is still a key issue in modern immunology. So historically, the self-tolerance has been studied and by various ways. The three mechanisms have been proposed. One is deletion. So dangerous self-reactive T cells or B cells, when they recognize self at their immature state, they'll die by apoptosis. It is called deletion or clonal deletion. A second mechanism is they recognize self-antigen, but then they don't die, but they just become inactivated. It is called anergy in immunological term. And third mechanism, that is, we do have the normal individuals, they possess a potentially hazardous self-reactive lymphocyte, but their expansion or activation, it is the actively suppressed another population of lymphocyte called regulatory T cells. So suppression. So deletion, inactivation, or suppression. So key question, and then what is challenging to immunologists is that which mechanism, if it's become defective, the autoimmune disease happens. In other words, how we can induce autoimmune disease by manipulating which one, when we manipulate them, we can induce autoimmune disease in normal animals. So here are autoimmune diseases. There are many autoimmune diseases. It is estimated that at least 5% of the population is afflicted with some sort of autoimmune diseases. For example, multiple sclerosis of the nervous system, thyroiditis, or gastritis, or the adrenalitis called Addison's disease, or type 1 diabetes. And then the systemic autoimmune diseases, such as SLE or rheumatoid arthritis. So currently, the cause and the mechanism of autoimmune diseases, they are still controversial. And one characteristic of these autoimmune diseases, that is overlapping. So if you see the patient with type 1 diabetes, he or she may also have thyroiditis or gastritis or other autoimmune diseases. So this type of the overlapping is very common in humans. It is also common in animals. For example, NOD mice, which is a good model of type 1 diabetes. But when they become aged, they develop also thyroiditis. And BB rats, it is also a good model of type 1 diabetes, but then they also, at the same time, develop thyroiditis and gastritis. And always strain chicken, it's a good model of thyroiditis, but then sometimes you see gastritis, autoimmune gastritis. So these findings suggest to us the two questions. One is, is autoimmune disease due to anomaly of the immune system or target cell functions. So historically, the autoimmune disease has been thought to be caused by, say, the viral infection of target antigen, target organs, and somehow self-antigen presented abnormally, and so the immune system start to react with self-antigens. And then second issue is the mechanisms of the immunological self-tolerance evolutionarily conserved, as shown here, animals and also humans. To address this question, there are two experimental systems. Both systems in mice and rats, just manipulating the immune system, you can induce autoimmunity. For example, Nishizuka and Sakakura, two Japanese pathologists, they reported in 1969 that if you remove the thymus around day 3 after birth, and somehow they developed autoimmune gastritis, ophlitis, and thyroiditis. And then also a group in the UK, Penn Hale, 1973, they reported that if remove the thymus of normal rats and then X-irradiated to gray every 2 weeks, and then some of them developed autoimmune thyroiditis and type 1 diabetes. So just manipulating the immune system, not the target antigen, you can induce autoimmune disease just similar to what we see in humans. So this is the neonatal thymectomy system. The bulb C, A, and C57 black mice, all are normal strains of mice, no autoimmunity. But if you remove the thymus around day 3 after birth, so 30% developed gastritis and also 25% ophlitis. But importantly, day 0, that is within 24 hours after birth, and you thymectomize, but nothing, almost nothing happens. And day 7 thymectomy, also you don't see any autoimmunity. And if you change the strain to A strain mice, then 90% develop ophlitis, autoimmune disease in ovaries, and then other autoimmune diseases as well. But C57 black mice, they are really resistant to this kind of autoimmunity. So the critical time of thymectomy, that is important. And also genetic background of the mice, that's also determine what kind of autoimmunity happens or doesn't happen. So important finding is then, this autoimmunity can be prevented by inoculating normal T cells from normal mice. For example, day 3 thymectomy can induce a 90% incidence of ophlitis, autoimmune disease in ovaries. But if you inoculate 5 million T cells from normal mice before thymectomy, or 1 week after thymectomy, or 2 weeks, you can completely prevent the disease development. So 3 weeks later or 4 weeks later, now mice become bigger, and then the 5 million T cells not enough, and so you'll see some autoimmunity to happen. So how we can interpret these findings? So thymus, it is producing T cells from gestational days on. So day 0, the mice are born. So day 3 thymectomy. So if you postulate that a thymus is producing T cells including hazardous autoimmune T cells, but somehow around day 3, they start to produce the kind of suppressive T cells. Now we call them regulatory T cells. So from the T cells which have already peripheralized, including autoimmune T cells, without regulatory T cells by thymectomy, they become expanded. They become activated and they expand and they cause autoimmune disease. So if you inoculate normal T cells, including regulatory T cells, after thymectomy, you can prevent disease. Then how we can demonstrate this mechanism more directly? So that is, you can just differentiate between regulatory T cells and autoimmune T cells in normal mice without thymectomy, and then directly if you remove them, whether you can induce autoimmunity or not. That is a key question. So basically, on this slide, the experiment we and others conducted in the past. So here, normal mice, the spleen, lots of T cells there. You can enrich CD4 T cells, and then remove the certain population by postulating that population may include regulatory T cells, and then transfer the remaining cells into T cell deficient mice, in this case, nude mice, congenitally deficient of the thymus. And then what we found, that we and others have found, is that if you remove certain population, and then they transfer the remaining cells. So these recipient mice, they spontaneously developed the various autoimmune diseases, just similar to human counterpart, and then inflammatory bowel disease as well. So what we did is, anyway, we removed the various population, and we removed this smaller number of population, a smaller size. And if we see more severe autoimmunity in a wider spectrum of organs, that is a specific population or specific definition of regulatory T cells. And we came up with CD25, that is IL-2 receptor alpha chain. So that's a good marker. So 10% of CD4 positive cells, they express CD25. And just removing this population, and then you'll see the various autoimmune diseases. So autoimmune gastritis, for example, the gastric mucosa is destroyed by inflammatory cell infiltration. You can demonstrate autoantibodies against the gastric parietal cells. This is actually the hydrogen potassium ATPase producing the hydrogen ion into the lumen. And just similar to the human pernicious anemia with autoimmune gastritis, thyroiditis with anti-thyroglobulin autoantibodies, and the type 1 diabetes. So you see the infiltration of inflammatory cells, and then islets, Langerhans islets are now being destroyed, and actually this mouse developed diabetes. So why day 3? So if you look at the ontogeny of CD25 positive CD4 T cells, so day 3 you see the CD3 positive, they are T cells, but not the CD25 positive cells. Day 7, you see some CD25 positive cells, and day 60, about 10% of CD4 T cells, they are CD25 positive cells. So this is ontogeny. In the spleen, the CD25 positive cells among T cells, you don't see them until day 3, but then after day 3, they rapidly, you'll find in the spleen, reaching to the adult level, 10% of CD4 positive cells. So neonatal day 3 in mice, thymectomy can deplete or reduce regulatory T cells and can cause autoimmunity. So day 3 in mice, it corresponds to neonatal day 3 in rats. So with rats, you must remove the thymus within 12 hours after birth. And then in humans, it is estimated to be around 12 weeks in gestation in utero. So based on these findings, now we know that the normal thymus is producing the regulatory T cells. So why day 3? So perhaps the regulatory T cells, they are produced by the thymus, and they differentiate into functionally competent regulatory T cells already within the thymus. So T cells are selected, the naive T cells, conventional T cells, and T regular, both are selected. But then it takes time for them to become the functionally competent cells. So that difference, time difference, the cause, that is responsible for why day 3 can separate somehow the Treg and regulatory the autoimmune T cells. So whatever it is, if the environmental or the genetic anomalies or environmental insult, when they affect the balance between Treg and the conventional T cells, including autoimmune T cells, that can be cause of autoimmune disease. So genetical anomalies affecting Treg or the environmental substances for certain viruses, for example, HIV virus for AIDS, the CD25 positive cells, they are firstly affected. So you see sometimes autoimmunity in AIDS patients. So cyclosporine A, so it reduces IL-2, and then CD25 is an IL-2 receptor. So without IL-2, the Treg cannot survive. And so you'll see autoimmunity. And I already mentioned to you, ionizing radiation. And so Tregs are more proliferative than other cells. So they are radiosensitive. So you can induce by these maneuvers, the induce autoimmunity by these treatment. And let me talk about genetic anomalies. Most important one is FOXP3. This is because there is a disease called IPEX syndrome, immune dysregulation, polyendocrinopathy, enteropathy, X-linked syndrome. And so the various autoimmune diseases in endocrine organs, including type 1 diabetes and enteropathy, inflammatory bowel disease. So autoimmune disease, especially type 1 diabetes, they develop in 80%, more than 80% of patients, thyroiditis 70%, within 3 years after birth. And then inflammatory bowel disease, almost 100%. So the milk feeding start to babies, and then immediately you'll see immune responses against commensal bacteria in the intestine. And allergy, so food allergy especially, it also happens almost in the 100% of the patients with hyper IgE in the circulation. So there is a counterpart in mice called scurvy mice. Similarly, it has the gene mutation on X chromosome. So it was first early around the year 2000. This gene was positionally cloned and then called FOXP3, the transcription factor. And then immediately it was found that this gene, FOXP3 mutation, is responsible for human IPEX syndrome. So here we made the antibody against FOXP3, and then you see the 10% of CD4 T cells, they express FOXP3, but in scurvy mice, no FOXP3 positive cells. And if you co-stain for CD25 as a marker for regulatory T cells and FOXP3 intracellular marker, then what you see is that a majority of CD25 positive cells, a majority of FOXP3 positive cells, they are CD25 positive in the spleen, also in the thymus. So in the past, we removed the CD25 positive cells, and we could see the autoimmune diseases. So this means we remove the majority of FOXP3 positive cells. If you remove all of the FOXP3 positive cells, you'll see more severe autoimmunity, and the mice will die because of systemic autoimmune diseases. So right now, FOXP3 and the CD25 is still the best markers for human regulatory T cells, not only in mice. So FOXP3, it is the just mutation of FOXP3 can be causative of various autoimmune diseases and also inflammatory bowel disease and allergy. And what function of FOXP3? So if you express FOXP3 in normal conventional T cells by retroviral transduction, and then FOXP3 transduced T cells, they can prevent disease development. So here, the Treg remove the T cells into skid mice, and you see colitis, inflammatory bowel disease, and also autoimmune gastritis. But if you co-transfer FOXP3 expressed conventional T cells, you can clearly prevent disease development. So here, normal colon, colitis can be prevented by FOXP3 transduced T cells. normal gastric mucosa, autoimmune gastritis, and gastritis can be prevented by the FOXP3 expressing conventional T cells. So these findings all together, what is important is, so normal individuals, they do possess potentially hazardous self-reactive T cells, but they are controlled not to be activated. But if you remove the Treg, and what happened is that common autoimmune diseases such as type 1 diabetes and thyroiditis, they develop, and also the magnitude against the commensal bacteria, it's increased. So you see inflammatory bowel disease, immunopathological disease, and also the Treg removed individuals, they start to react with the environmental substances, allergy. So the border between self and non-self, it can be movable. So if you further down, you may see autoimmunity, but you may see also a good tumor immunity. But on the other hand, if you raise this threshold, you can induce a transplantation tolerance. So here, tumor immunity, the same experimental system, T-cell deficient mice, if you transfer 4 T cells and inoculate the tumor cells, tumor become bigger, and then mice will die by tumor progression. But if you remove Treg and then transfer to T-cell deficient mice, they do develop autoimmunity. But at the same time, they show a very strong the immune reaction responses against tumor cells. So tumor become bigger and then rejected. And after rejection, if you inoculate 10 times the number of tumor cells, immediately they are rejected, meaning that they became immune to the particular tumors. So if you then mix 1-to-1 ratio, now the Treg become half, so 4.4% to 2.5%. But still they suppress immune response, so tumor become bigger, and then mice will die by tumor progression. How about transplantation? Again, the T-cell deficient mice, nude mice. So just you can transplant the skin from other mice, although it can be accepted because these mice, they don't have T cells. But if you transfer naive T cells from the syngeneic mice, and then they start to reject the skin graft. So within a month, all the transplant, the grafts are all rejected. But if you first transfer Treg, and in 1 week, so the transfer Treg, they are recruited to the graft site, and then suppress immune response. So the rejection becomes delayed, and 30% show the permanent tolerance, accepted. And if you increase the Treg to, say, 1-to-3 ratio, now 70% of them now permanently accept the allogeneic allograft. So just the targeting Treg without any immunosuppressive drug, you can induce transplantation tolerance. So this is our current understanding. So the normal thymus, the highly self-reactive T cells, they are deleted. But potentially hazardous self-reactive T cells, they are produced, and then they persist in the periphery. But the thymus also produces the regulatory T cells expressing FOXP3, and then they actively suppress the expansion of the autoimmune T cells, preventing autoimmune disease, and then also suppressing allergy or inflammatory bowel disease. But also by reducing them, you can enhance tumor immunity, and then by expanding them, you can induce organ transplantation. And also fetal-maternal tolerance. Why the gestation, the pregnancy can continue because the mother harbors the non-self fetus, but the mothers, they don't show rejection. This is called fetal-maternal tolerance. And then you see in the uterus and placenta, you'll see a lot of Tregs are recruited in early phase of pregnancy. And also recently, the obesity or other diseases, immunometabolic diseases. It accompanies inflammation. And then inflammation site, for example, in fat tissue, Tregs are really recruited and then controlling the inflammation. So thymus produces Treg, and they are expressing CD25, and then also they are also expressing CTLA4, another cell surface marker. And some of them in the periphery, especially in the intestine, the naive T cells can develop into Treg in the mucosal tissue. They are called P-Treg, thymus-derived T-Treg, peripherally derived P-Treg. And then I-Treg means in vitro induced Tregs. So let me talk about the function of regulatory T cells. So they are expressing CD25, CTLA4, and then recently it was found that their expression of PD-1 is important for their function. So FOXP3, transcription factor, it controls the hundreds of genes. It binds to the promoter region of at least 100 genes. So you can ask which gene downstream of FOXP3, which one is important? So you can ask then which gene downstream, if you make knockout mice, whether they develop similar autoimmune diseases as FOXP3 mutation, such as scurvy mice or IPEX syndrome in humans. There are not so many. So IL-2 knockout, or CD25 or CD122. CD122 is the IL-2 receptor beta chain. These knockout mice, they spontaneously develop the very severe autoimmunity, like FOXP3 mutation. So another one is CTLA4. So let me talk about IL-2. So FOXP3, the key feature of function of FOXP3 is repression of IL-2. So the Treg, they don't produce IL-2, but they do express the high IL-2 receptor. So IL-2 receptor alpha chain, this is CD25, and the beta chain, gamma chain all together, they formed the high affinity IL-2 receptor. So the key feature of Treg, they are constitutively expressing the high affinity IL-2 receptor. So they can easily bind IL-2, but they themselves, they don't produce IL-2. So that is a big difference between Treg and then activated T cells. So if neutralize IL-2 from the body by just injecting neutralizing anti-IL-2 antibody, so what you see is here. So Treg in the spleen has CD25 positive cells, also in the thymus. By just one injection of IL-2, neutralization, then Treg will die. And then as a result, you can induce autoimmune disease. So day 20 after birth, just inoculating neutralizing IL-2 antibody, that's enough for inducing autoimmunity. So this is normal bulb C mice, and you see the autoimmune gastritis with a high titer of anti-parietal cell autoantibodies. This is NOD mice. They spontaneously develop the diabetes, but not so severe. But if you inoculate anti-IL-2, the islet, the insulitis in the pancreas become severe, and then thyroiditis more severe, forming the goiter, and salivary gland and lacrimal gland adenitis. So there are many cytokines, but only cytokine which can induce autoimmunity, that is IL-2. Just neutralization of IL-2 is enough for inducing autoimmunity by affecting Treg. So then CTLA4. So similarly, if you neutralize the block CTLA4 by anti-CTLA4 antibody just twice, day 20, and 3 months later, you'll see a very severe autoimmune gastritis on the bulb C background. So just similar to IL-2 neutralization. And then if you make the Treg specific CTLA4 deficient mice, so here FOXP3 positive cells, they don't express CTLA4, but in other activated T cells, they can express CTLA4. So normally the FOXP3 positive cells, they're highly expressing CTLA4, but the knockout mice, they don't. And these mice, spontaneously develop very severe autoimmune diseases, such as autoimmune myocarditis. So here you see it's ventricle, and then the blue parts are all infiltration of inflammatory cells. So this is atrium. You see the destruction of cardiac myocytes. And they show heart failure, cardiomegaly, and they'll die. And if you look at the IgE, measured IgE in the circulation, several hundred-fold higher level of IgE, so they are prone to develop allergy. And then, as you know, CTLA4 antibody, it is used for the anti-cancer immunity. So if you inoculate the tumor cells, so red one, knockout mice, first the tumor become bigger and then rejected. But the control mice, they all die of tumor progression. So just similar to Treg depletion, just with the non-expression of CTLA4 specifically in Treg, then you'll see the autoimmunity, allergy, and good tumor immunity. In humans, there are the CTLA4 mutations. So these two patients, they are heterogeneous, heteroinsufficiency. So compared with the control individual, after activation, FOXP3 positive cells, they express CTLA4, but these patients, they cannot upregulate the CTLA4 expression. And then they do develop very severe autoimmunity and immunopathology. So what's the role of CTLA4 for regulatory T cells? So this is in vitro mixed lymphocyte reaction. This is alloantigen stimulation. And then the red one is a knockout, a CTLA4 knockout Treg. So normal Treg, so responder T cells, they can respond. And then if you add the regulatory T cells, then the regulatory T cells suppress the proliferation of responder T cells, 1:3 or 1:1 ratio. But red one, knockout Treg, they don't show good suppressive activity. And how? So here the APC and Treg, and then the CD86 blue and CTLA4 red. And what happens in co-culture and activated? So by interacting with Treg interaction with APC, the green one become aggregated on the cell surface of antigen-presenting cells. And then they transfer to Treg. It is called trogocytosis. And then they are the endocytosed into Treg, called transendocytosis. As a result, you'll see the reduction of the CD86 co-stimulatory molecule on the cell surface of APCs. So here, activated DCs, they are expressing CD80, and then also PDL1 here. And then after the co-culture with Treg, and as you don't see, but the CD80 expression become lower. And importantly, if you stay in for free PDL1, and then usually it's a small number of the dendritic cells express free PDL1. But now with incubation Treg, free PDL1 and also CD80 low population, they become they constitute a major population. So the interpretation is here. So in normal state, CD80 and the PDL1 is cis-associated. And then Treg comes and interact with the CD80 and then remove CD80 as shown here. And then the PDL1 become free. And actually these free PDL1, they can interact with PD1 expressing T cells. And so they inhibit PD1 positive effector T cells. So this means that by Treg, the CD80-86 downregulated, meaning that the naive T cells cannot be activated. And at the same time, the increase in free PDL1, they engaged with the PD1 positive T cells and suppress their function. So all these findings together, the FOXP3 repress IL-2 production, upregulate IL-2 receptor, and so they can deprive IL-2 from surroundings. And also, they upregulate CTLA4 and CD80-86 downregulation. And as a result, the responder T cells, they can recognize T cells, the antigen, but without signal 2 co-stimulation. Then, depending on the TCR affinity for the MSC, peptide MSC, if it's the high affinity, without co-stimulation, they'll die by apoptosis. If it's intermediate, they become inactivated, called anergy. If it's a low affinity, they stay dormant. And also, CD80-86 downregulation upregulate free PD-L1, and the PD-1 expressing the T cells also suppressed. So in tumor immunity, in cancer immunology, anti-PD-L1 and nivolumab, anti-CTLA4, ipilimumab, they are really potent in tumor immunity. But at the same time, they can induce, they'll induce the autoimmunity and also the inflammatory bowel disease. So mechanism is this. So here, if you block here CTLA4, then downstream events, so they cannot be suppressed. So you'll see autoimmunity and the tumor immunity. And the PD-1 blockade here, it's less severe than CTLA4 blockade, but still you see good the tumor immunity and at the same time autoimmunity. And the Treg also further activated, they produce IL-10 or other immunosuppressive substances and then control APC function, especially CD80-86 expression. So now, based on these findings in mice and humans, let me talk about human autoimmunity. So here, since the discovery of regulatory T cells, so lots of effort have been done to look at any anomalies in autoimmune disease patients. So you can see some differences, but you cannot tell whether this is the cause or consequence of the disease. So we decided to look at the single nucleotide polymorphism of GWAS. So any the GWAS SNPs, whether they affect genetic control of a Treg function development, and thereby contributing to determining genetic susceptibility to autoimmune diseases. So the clue is super enhancer. So in the genome, here a typical enhancer, and then the enhancer controls the promoter region and the gene expression. But then there are many very strong enhancer regions called super enhancers, where you see a dense accumulation of transcription factors and the activation of histones, for example, H3K27 acetylation, and then CBP or P100 binding. So these super enhancers, now it has been shown that it controls the cell identity and the functional stability. So very strongly control the downstream genes. So this means that the Treg-specific super enhancers may determine the Treg cell fate and then also provide confer functional stability. So this is the Treg, and then here a CD28 gene and the CTLA4. So if you compare with conventional T cells, so this is H3K27 acetylation. This is a marker for active enhancer. So you don't see much difference. This is called common super enhancer. But if you look at the CTLA4, Treg highly activated compared with the conventional T cells. It is called a Treg-specific super enhancer. So in the genome, the Treg-specific super enhancers. So if you put the enhancers from a small one to large one, and then the ordinate is H3K27 acetylation, the intensity. So it's not a linear increase. At a certain point, then all of a sudden it increases. So this region is called the definition of super enhancers. And you see the Treg really possess the super enhancers at the CTLA4 gene, FOXP3, IKZF2, this is Helios, and also IL-2 receptor alpha chain, CD25. So we found Treg-specific super enhancers, 66, a common one, around 300, and conventional T-cell specific super enhancers, around 40. So by tracing super enhancer activation, you can analyze the Treg differentiation. So in the thymus, CD4, CD8 double positive cells become immature CD4 single positive, and then pre-Treg. Pre-Treg means CD25 positive, but FOXP3 negative, and thymus-derived T-Treg, FOXP3 positive, and CD25 positive. So what is interesting is here FOXP3 gene, red line, it is super enhancer. So even at very immature stage, now the histone activation, it starts and becomes stronger along Treg differentiation. Similarly, H3K4 monomethylation, this is a marker for a poised enhancer, just ready to be activated. And again, they become stronger along Treg differentiation. And also ATAC sequences. This is a marker, the indicator of chromatin remodeling, opening. So at the double positive stage, you see some change, and then this change becomes stronger along Treg differentiation. So this means that before FOXP3 expression, at the immature stage, already the Treg lineage is already determined. So that is important. And not only FOXP3, other CTLA4 or CD25 Treg signature genes, in parallel, they become activated along Treg differentiation. And then, so this is current understanding. So developing T cells, expressing T cell receptor, they interact with self-MSC the antigen, the ligands, and then somehow the developing T cells are committed to Treg. We don't know yet what kind of pioneer factor is involved. But as a result, super enhancer activation of various Treg signature genes, and then a gene expressed, and also at the same time, the Treg type epigenome is installed. And these two together, now we have a function competent, the Treg. So based on these findings in mice, now we can analyze a human peripheral Treg. So this is peripheral blood. So CD25 and FOXP3 is a good marker. You can define the FOXP3 positive, CD25 positive cells. So they are kind of proportional. But we also know that some FOXP3 positive cells among them, they are not Treg, just activated T cells. So FOXP3 is not strictly specific in humans. So question is how we can define genuine Treg. So just looking at the epigenome, so looking at the CpG hypomethylation between the mature Treg versus conventional T cells. So conventional T cells, they are methylated. But these regions, the Tregs are, the red means demethylated. And you see fraction 1 and fraction 2 is very similar. But the fraction 3 here, FOXP3 low and CD45RA low, they are more like conventional T cells. So meaning that fraction 1 and 2, they are the Treg lineage. So by analyzing super enhancer, Treg specific super enhancers in Treg lineage, fraction 1 and 2, so we found that 21 Treg specific super enhancers. And indeed, various autoimmune SNPs, they are enriched in Treg specific super enhancers, not in activation-induced super enhancer or common super enhancers. Here, vitiligo or Graves' disease or multiple sclerosis, the SNPs are highly enriched in the Treg specific super enhancers, type 1 diabetes and other diseases. So many autoimmune SNPs, they control the Treg function and development. So here, this is the old paper published by Wellcome Trust in 2007. Actually, this is the first paper of GBAS analysis. And then type 1 diabetes, so HLA, insulin, CTLA4, and IL-2 receptor alpha, CD25 here. But at that time, we couldn't tell whether these SNPs really affect the activated T cells or regulatory T cells. Now we know that these SNPs really they affect the Treg function and development. So let me summarize this part of my talk. So there are many monogenic autoimmune diseases affecting regulatory T cells. So now they are called T-regopathies. So mutation of Treg signature genes such as FOXP3, CD25, CTLA4 cause monogenic autoimmune diseases by affecting Treg development and function. And how about the common autoimmune diseases, polygenic ones? So the genetic polymorphism, in particular in Treg-specific CpG hypomethylation regions and super enhancers, such as FOXP3, CD25, CTLA4, mainly affects the function and development of endogenous naive Treg, and thereby contribute to determining genetic susceptibility to various polygenic common autoimmune diseases. So this is an interesting finding made by Jean-François Bach, an eminent immunologist in France. So he published many years ago that if you look at the incidence of infectious diseases and the incidence of various other the immunological diseases, so what you see here is that in developed countries, the incidence of the infectious disease is really decreasing. Of course, we suffered from COVID-19 the pandemic, but in general, common infectious disease is really decreasing. But on the other hand, the autoimmune diseases such as type 1 diabetes or multiple sclerosis it's increasing. Inflammatory bowel disease such as Crohn's disease or allergy, asthma, they are increasing. So these increasing immunological diseases, they are really we see in IPEX syndrome or by Treg depletion or dysfunction. So the question is, the Treg somehow involved in these, the increase in autoimmunity or immunopathology in hygienic societies in developed countries. So why infectious disease decreases, but an autoimmune disease is increasing? So one scheme is shown on this slide. So whatever it is, genetic factors or environmental factors, if it affects the Treg function, then you can see autoimmunity. But what kind of autoimmunity happens? It really depends on host gene, the background. So as already I've shown you that by neonatal thymectomy, depending on the mouse strain, you'll see different autoimmune diseases, just similar. So the depletion of Treg or reduction of Treg, these patients develop type 1 diabetes, but the second one developed thyroiditis and gastritis, but the third one, no autoimmune disease. And really, this pattern is depending on the host gene background, genetic background, MSC and non-MSC genes. So in developed countries, if the immune system is not much stimulated, because environmental, so viral infection, if you have a viral infection, tissue destroyed, and then self-antigen released, and then the regulatory T cell recruited, and they become activated. If it doesn't happen, then the regulatory T cells, they are not trained or become become strengthened. So the self-reactive T cells, dangerous ones, accumulate, but not enough. And then the stimulation of innate immunity, now you see overt autoimmunity. So type 1 diabetes patients, just a seasonal viral infection, that's enough as a final triggering of autoimmune disease. So this scheme may explain how autoimmune disease happens in general. So let me move on to the third part of my talk about how we can target regulatory T cells to treat or prevent autoimmune disease. So important one is antigen-specific immune suppression. So steroid or tacrolimus or cyclosporine A, we have a very good the immunosuppressant, but they are all antigen non-specific. So holy grail in transplantation immunology or immunology is how we can achieve antigen-specific immune suppression. So currently, as I already told you, that how we can expand Treg, low-dose IL-2, so Tregs are expressing high affinity IL-2 receptor. So you can expand Treg by low-dose IL-2, not affecting effector cells. So, and then other treatment, and then you can expand Treg, natural Treg. And also, you can take them out, and then ex vivo, stimulating them with antigen, and then expand them, and transfer back to the patient. So all these treatments now undergoing in Europe and in the United States. What we are doing is, well, ultimately, the ideal way of antigen-specific suppression is, so antigen-specific effector T cells here, mediating disease, if they can be converted to Treg in vivo or ex vivo, and then transfer back to the patient, that is the best way of the antigen-specific suppression. So to do that, we looked for any substance which can convert effector T cells into Treg. So we surveyed the library of the tyrosine kinase inhibitors or serine threonine kinase inhibitors, more than 5,000. And then just three-day culture, putting such substance into the in vitro culture with T cell stimulation, and whether FOXP3 can be induced or not. So one substance, this is not the natural one, and this is artificial one, AS286 and 3619. It is abbreviated as AS compound. So here, the naive T cells, CD44 low and CD62L high. So naive T cells, when they are stimulated with TGF-beta, you can see the FOXP3 positive cells. And here, by neutralizing TGF-beta and putting this AS substance, you can induce the FOXP3 positive cells. And more importantly, this population here, they are effector memory T cells. So TGF-beta cannot induce FOXP3, but this substance can induce FOXP3. And even from CD8 positive cells, you can induce the FOXP3 positive cells. And then this substance, under inflammatory conditions, for example, Th1 or Th17, so we control lots of interferon gamma secreting cells. And then if you put AS compound, now interferon gamma secreting cell reduced, but then FOXP3 positive cells, they increase. Similarly, Th17 cells reduced, FOXP3 positive cells induced. And also Th2 condition or Th9 condition. All these inflammatory conditions, you can induce the FOXP3 positive cells. And then ACE compound, how it works. And then the mass spectrometry, we found that this substance binds to CDK8-19. This is a regulatory module of a mediator complex which controls the transcription of the many genes. So if you knock down CDK8 or CDK19 in T cells, you see upregulation of FOXP3. Or kinase dead CDK8 and CDK19. So it's a negative, the dominant negative effect. So either one alone can induce FOXP3 positive cells. And they increase the STAT5 binding. So CNS0 region of FOXP3 gene, this is first activated, and then in the presence of AS compound, the STAT5 really bind to this region and activate FOXP3 genes. So here activated conventional T cells, the T cell receptor signal activated CDK8-19 and inhibits STAT5. And STAT5 is upregulated also IL-2 signal. And if you inhibit CDK8-19, this inhibition is gone, and STAT5 is activated and activate FOXP3 gene. So in vivo, the antigen stimulation plus this compound. So here we are using the over-albumin-specific T-cell receptor transgenic mice. So the AS compound cannot induce FOXP3 positive cells. But here, antigen plus AS compound, orally taken, and now you can induce the antigen-specific Treg, as shown here. They are similar to natural Treg, and also epigenome, demethylation of FOXP3 gene or other Treg signature genes, all demethylated, meaning that they are functionally stable. So the contact hypersensitivity, we treat with AS compound orally, you can prevent the contact hypersensitivity. But if you remove Treg, you don't see such effect. So AS compound increase Treg, induce Treg, and then suppress contact hypersensitivity. And NOD mice, just for a limited period, orally taken, and then just as the early phase of type 1 diabetes, and now you can prevent disease development. So histologically, majority of islets, they are intact. So currently, we plan to do clinical trials by this kind of approach. So the memory effector T cells mediating autoimmune disease or allograft rejection or allergy. So whatever it is, we know sometimes the target antigen, but in a majority of cases, we don't know the target antigen. But still, effector memory T cell fraction, they can be converted to FOXP3 positive cells. And then we can use them for a treatment in vivo conversion as in vivo conversion or in vitro conversion for cell therapy. So this is the last slide of my talk. So I have mainly talked about how FOXP3 positive cells can be targeted to treat the various immunological diseases, including autoimmunity, allergy, IBD, organ transplantation, or other physiological conditions. On the other hand, by reducing Treg, you can enhance the immune responses like tumor immunity or chronic microviral infection. So this part is just easier than Treg up therapy, just reducing them, whatever it is, by antibody or small molecules, you can reduce them, and then you can enhance the tumor immunity. So we do hope that in the future, in near future, so by targeting Treg, either expanding them or reducing them, we can control physiological and pathological immune responses in the clinic. So many people, the students and postdoc have been involved in these studies. I really appreciate their contribution. Without them, I'm not here to be awarded with this Debrecen Award for Molecular Medicine. And we have many collaborators, clinicians, and also the scientists in other fields. So I'd like to acknowledge them. Thank you very much for your attention.
Professor Sakaguchi, thank you very much for accepting our invitation and for your very thrilling lecture. We wish you continued success in your personal life and in your academic life. Thereby, I declare this ceremony closed. Thank you.